In this video, you'll learn a protocol for testing enzyme activity, so make sure you're setting up your lab notebook as you go. Just a little background information. In this lab, we're going to be working with a particular chemical reaction that we've been studying in class. That chemical reaction is the hydrolysis of starch. So in our lab, starch is going to be the substrate. Remember that starch is a carbohydrate and it's a polymer made up of smaller molecules. The enzyme that we'll be using in lab is amylase. Amylase is an enzyme with an active site specific for starch only. And glucose is the carbohydrate monomer that makes up starch. So that is going to be the product at the end of the reaction. Now, as we're testing enzyme activity, we need a way to know if the reaction occurs or not. So we're going to use indicators, specifically Lugol's solution. Lugol's solution will turn from orange-brown, its natural color, to black in the presence of starch. So, the lighter the solution, the less starch present and the more enzyme activity. If you look at these various solutions, you can see that these solutions are very light. That means that a lot of hydrolysis occurred, there's no starch left, and we can conclude that there's high enzyme activity. The opposite, the darker the solution, the more starch present and the less enzyme activity. So we can see in this well on the spot plate, because it's very black, there must be a lot of starch present, which means not very much reaction occurred. And thus, there must not be very much enzyme activity. So the problem we're investigating in this lab is, how do various environmental conditions affect enzyme activity? And those environmental conditions could be temperature or pH. We're also going to take a look at how the concentration of enzyme affects enzyme activity. So in this lab, our independent variable, what we're going to be changing, is the environmental condition, and the dependent variable, what we will measure, is enzyme activity. Take a moment right now and write a quick hypothesis for each of those environmental conditions. How do you think enzyme concentration will affect enzyme activity? How will changing the temperature affect enzyme activity? And how will changing the pH affect enzyme activity? All right, now let's take a look at how we're going to conduct this lab. You're going to be using spot plates, just like in our last lab, and you're going to use masking tape to label the columns 1 through 4 and the rows A, B, and C. Now, the first experiment we're going to do is for enzyme concentration, and we're going to use row A to test enzyme concentration. So in the first well, you're going to add 3 drops of water and no enzyme. This will essentially serve as your control. In the next well, you'll add 2 drops of water, 1 drop of enzyme, 1 drop of water, 2 drops of enzyme, and then finally 3 drops of enzyme in the last well. What we're doing here is we're keeping the total amount of solution constant in each well, but we're changing the concentration of the enzyme. Then we need to add our substrate. So to each well, as quickly as possible, you're going to add 7 drops of starch. You want to add the starch as quickly as possible because you want to give each well the same amount of time to react. So you'll wait 30 seconds after adding the starch to give time for hydrolysis to occur. After 30 seconds, you're going to add a drop of Lugol's solution to each of the wells. And you can shake the tray gently to mix the solution, but as soon as it's mixed, record the color of the solution in each well. Again, pay close attention to time. This is our basic protocol. Now you're going to modify that protocol to test the effect of temperature on enzyme activity. So you're going to use row B to test temperature. And there's going to be enzyme solutions available at different temperatures. There'll be an enzyme solution on ice, an enzyme solution in a warm water bath, and an enzyme solution in a hot water bath. It's up to you exactly how you want to carry out the experiment. Just keep in mind that you want to keep the time and the total amount of solution in each well constant. The only thing you want to change is the temperature for row B. And then finally, you're going to use row C to conduct an investigation on pH. There will be hydrochloric acid and sodium hydroxide, a base available in the lab. Again, you can conduct the experiment how you wish, but 3 drops is a recommended amount of acid and base, and make sure that you keep time and the total amount of solution in each well constant. Once you've done those 3 experiments, you're going to wash and dry your spot plates thoroughly. And your results can be recorded in a data table like this one. So this data table will work for row A as you're testing enzyme concentration. Note, because the total amount of drops is 10 drops, we can convert this into a decimal to measure concentration. So 3 drops out of 10, 2 drops out of 10, 1 drop out of 10, and 0 drop out of 10. Here you'll put the color of the solution, and here you want to give a qualitative estimate of the enzyme activity. So if the solution is black, you might say the enzyme activity is 0, or use a minus sign. If the color of the solution is a dark brown but not completely black, you might want to say that the enzyme activity is low, or give it 1+. If the color of the enzyme solution is a really light orange, then you could say +++, or high for enzyme activity. Your lab group will have to make another table for temperature and a third table for pH, and it's up to you how you design that table. Finally, for your analysis and conclusions, as always, you're going to write a claim for your temperature investigation, provide evidence, and then justify it. And you may not have a good idea of this until our lesson on Friday, but that's okay. Then you'll write a claim for the pH, evidence for the pH investigation, and a justification. Finally, evaluate your protocol. Were there any sources of error? Did you use too many drops? Were you sloppy about the time? And what's a new question you'd like to investigate that relates to enzyme activity? And now you are all set to conduct our lab on enzymes.